Hello, my name is David Merrell. I'm a PhD student at the University of Wisconsin. I'm also with the Morgridge Institute for Research. I'm advised by Anthony Gitter. Today I'm presenting work entitled Inferring Signaling Pathways with Probabilistic Programming. So some motivation to start with. We're trying to solve a problem called signaling pathway inference. The idea is that cells regulate themselves via complicated biochemical processes called signaling pathways. You can think of a signaling pathway as a directed graph where the nodes are proteins and the edges are regulatory relationships between them. We want to infer signaling pathways from time series phosphorylation data. So that's our problem. How do we solve it? We formulate the problem as a dynamic Bayesian network structure estimation problem, and we take a Bayesian approach. We want a posterior distribution over possible graph structures, conditioned on the time series data. We decompose this posterior distribution into a graph prior and a graph likelihood function. We accept prior knowledge in the form of a weighted directed graph, which in practice we can derive this from, say, signaling pathway databases, KEGG for example. The graph likelihood function is a dynamic Bayesian network. The structure of the dynamic Bayes net is determined by the graph structure. So that's how the time series data are connected to the graph structure. The product of these two things gives us an unnormalized posterior distribution. But we still need a way to use this unnormalized posterior. We need an algorithm to compute posterior probabilities from it. To do that, we designed a method called Sparse Signaling Pathway Sampling, or SSPS for short. SSPS is a Markov chain Monte Carlo method. It samples from this posterior. It goes on a random walk through the space of directed graphs. Our biggest contribution here is that we invented a proposal distribution that efficiently explores that space. And it's different in that it operates on the parent sets of the nodes, of the graph nodes. At each step of the Markov chain, it proposes one of three moves. It either adds a parent to a vertex, removes a parent from a vertex, or it swaps a parent, which is just a simultaneous application of add and remove. The intuition is that when a node has too few parents, we give a high probability to the add parent move. When it has too many parents, then we give high probability to the remove parent move. And when the number of parents is about right, we give high probability to swap parent. Assigning probabilities like this biases the exploration towards sparse graphs and yields efficient sampling. So that's SSPS. We implemented SSPS as a probabilistic program in Julia using the GEN probabilistic programming language. Probabilistic programming is a methodology for building modular, reusable, and extensible models. You can find all of our code on GitHub. We put a lot of effort into reproducibility. We implemented all of our analyses as a snake-make workflow. So definitely check it out if you're interested. GitterLab/SSPS. So all of that sounds good, but we still haven't talked about whether SSPS actually works. We answered that question by evaluating SSPS on real and simulated data. The simulation study is useful because it allowed us to see whether SSPS can actually identify the true network in a setting where we have access to the ground truth. And what we found is that SSPS did an excellent job of recovering the true network, even when the prior knowledge was corrupted. It was also more efficient than other dynamic Bayes net-based techniques. We also evaluated SSPS on empirical data, experimental data, from a dream challenge from a few years back. The short story of it is that the dream challenge had these 32 biological contexts, and the goal was to infer causal molecular relationships in those contexts. The takeaway is that SSPS performed about as well as existing methods on this task. However, due to some issues in the design of the challenge, it's hard to say anything much stronger than that. So, bottom line, SSPS appears to be a viable method. And it certainly is an advance in the area of dynamic Bayes net approaches to this problem. To close, I'll mention that I'll acknowledge our funding from the National Institutes of Health and from the National Science Foundation. I will also mention that our manuscript was accepted for publication in the ISCB 2020 conference proceedings. For now, you can find it on archive under the same title as this slide, or as this poster. So with that, I'm finished, and I welcome any questions.